Okay, so welcome to this next video in which we are discussing high-density lipoproteins. Okay, right. So in this video, what we now want to look at is how HDL and LDL lipoproteins are going to deliver their cholesterol content to cells, okay? And we've seen the main two categories of cells that HDL and LDL particles are going to deliver their cholesterol to are hepatocytes, okay, so cells of the liver which will use the cholesterol to make bile, okay, and also steroidogenic cells such as the cells of the adrenal cortex and the cells of the gonads, which will use cholesterol to make steroid hormones. Okay, right, we're going to concentrate on hepatocytes, okay, and we're going to see how HDL and LDL particles will deliver their cholesterol esters to the hepatocytes, and they do it in very different ways. So let's start off by looking at high-density lipoprotein. Okay, so let's draw our hepatocyte here. Okay, so this box will represent our liver cell. Okay, so this is a hepatocyte, and on the surface of the hepatocyte you will have a receptor for HDL. Okay, and this receptor is not called the high-density lipoprotein receptor, but instead is called the scavenger receptor B1. Okay, and for short, the scavenger receptor B1 is often abbreviated to the SRB1. Okay, so scavenger receptor B1 or SRB1. Alright, so let's colour in the scavenger receptor B1 then in pink here. Okay, and this is going to bind to the HDL particle, and what's going to happen is the cholesterol esters that are within the spherical HDL particle are going to be transferred out of the HDL particle and into the hepatocyte. Okay, so let me draw my HDL particle here. Okay, so here's our HDL particle, and the scavenger receptor B1 will interact with the apolipoprotein A1, which is in the phospholipid monolayer of our HDL particle. Okay, so here is apolipoprotein A1 here in turquoise. Okay, and remember that our phospholipid monolayer contains a lot of lecithin molecules with some cholesterol molecules distributed in there as well. Okay, but the main thing we're going to transfer out of the HDL particle into the hepatocyte will be the cholesterol esters, which are in the core of the HDL particle. So these cholesterol esters are going to go from the HDL particle into the hepatocyte. Now, note, the HDL particle is not endocytosed. This is in contrast to the LDL particle, which is going to be completely internalized by the hepatocyte and broken down. In the case of HDL, high-density lipoprotein, what happens is the HDL docks at the hepatocyte and then empties basically into the hepatocyte, so it gives all of its cholesterol esters, which are in the core, to the hepatocyte. And what does that then result in? Well, it results in the HDL particle going back to being a discoidal HDL particle with no hydrophobic core. Okay, so once it's given up all of its cholesterol esters that are within the core, it returns to this discoidal structure, and these discoidal structures will then go back into the blood, and they can then go round to other cells which are overloaded with cholesterol and take cholesterol and lecithin from those cells and replenish their cholesterol ester core, and then they can come back to the hepatocytes and deliver it to the hepatocytes. Okay, and this is the continuation of that reverse cholesterol transport process. Okay, so we recycle the HDL particles basically by turning them back from being the spherical HDL here, which is the mature form of HDL once it's gone and received a large amount of lipid molecules from overloaded cells, and we return them back to this nascent discoidal form. Okay, so this is a nascent discoidal HDL, and these discoidal HDLs can then go back round for another cycle, basically. Okay, so that's how we get the cholesterol esters out of HDL particles. Let's contrast this now to what happens in the case of LDL, okay? Because LDL is going to be completely endocytosed when it binds to its receptor. So, let's put the LDL receptor here on our hepatocyte. Okay, so this can represent the LDL receptor. So this is the receptor for low-density lipoprotein that's often just abbreviated to the LDLR for low-density lipoprotein receptor. Okay, so I'm going to colour this in green here. Okay, and the Ldl binding domain of the LDL receptor, it has a special name, so we'll represent this as this sort of flat portion on the extracellular side of the membrane. It's known as the type A repeat domain. Okay, so this is the type A repeat domain, and this is the portion which will bind to the LDL particle, specifically the apolipoprotein B100 within the LDL particle. Okay, so let's now show this process. So let's have our LDL receptor redrawn out here with this type A repeat domain shown here. And what's now going to happen is an LDL particle is going to come along, and remember, these things too are going to be stuffed full of cholesterol esters. Okay, so here is our LDL particle, like so. And then we've got our large apolipoprotein B100 in the phospholipid monolayer, and this is the structure which will bind to the type A repeat domain of the LDL receptor. Okay, so here in orange, this represents the apolipoprotein B100. So this is apo B100. Okay, and then we know that the phospholipid monolayer then consists of many lecithin molecules, like so. Okay, and cholesterol molecules will also be within the phospholipid monolayer. And then in the center of the LDL particle, you've then got cholesterol esters. So you've got this large store of cholesterol esters in the core of the LDL particle. And some of these will have come from HDL particles which have passed the cholesterol esters onto LDL particles. Okay, now, unlike the HDL, the LDL isn't recycled, okay? So instead what's going to happen is the whole thing is going to be endocytosed into a little endocytic vesicle. So let's discuss this in a little bit more detail. So basically, let's start by discussing the interaction between the type A repeat domain and then also the apolipoprotein B100. So basically, in the type A repeat domain of the LDL receptor, you find a lot of acidic residues. Okay, so many acidic residues are present within the type A repeat domain of the LDL receptor. Whilst in the apolipoprotein B100, you find a lot of basic residues. Okay, so how are these important in the interaction between the type A repeat domain of the LDL receptor and the LDL receptor's apolipoprotein B100 here? How is that important, basically? Okay, well, basically, if I show you an acidic residue and then a basic residue, we might be able to understand how this is going to help these two proteins interact. So let me start by showing you an acidic residue. So the acidic residue I'll use will be aspartic acid. So here's the amino terminus, here is the alpha carbon with a hydrogen coming off, and here is the carboxylic acid terminus linked to the amino terminus of the next protein along. So I'm drawing this amino acid as though it's an amino acid residue within a protein, rather than just the free amino acid. Okay, and then the R group of aspartic acid consists of a methylene group, like so, and then a carboxylic acid group. Okay, so this is aspartic acid. Okay, now, in physiological pH, what will generally happen to acidic amino acid residues, such as aspartic acid, is that they will lose their protons from the oxygen of the alcohol group within the carboxylic acid group. Okay, so the proton will dissociate off, leaving this oxygen with a negative charge, and the conjugate base of aspartic acid is called aspartate. Okay, so most of the acidic amino acid residues will exist as their conjugate bases rather than as the actual acid molecule. And remember, it's called a conjugate base because it's no longer an acid, because it can't donate protons away anymore. Instead, it's actually a base because it can receive protons onto that negatively charged oxygen. Okay, right, so a good fraction of the acidic residues which are in this type A repeat domain of the LDL receptor will be negatively charged because they'll have lost their protons, and the others will just be neutral. Then, if I show you a basic residue, and the sort of archetypal example of a basic residue would be lysine, so I'll show you lysine. So again, here is the core amino acid structure now, and then the R group of lysine consists of 4 methylene groups, which I'll just abbreviate like this. I'll draw one methylene group, and then I'll use my trick of putting brackets around there and subscripting it 4. And then we've got an amino group right on the end. So this is a lysine amino acid. Now, that nitrogen in the amino group has a lone pair of electrons, okay, which is a nice center of negative charge here. So I'll circle it. Okay, and what can happen is protons in free solution can come and associate with that center of negative charge. So you can bring in your proton here and it can associate with that. And that will give the entire molecule a positive charge now. So many basic residues will exist as their conjugate acids in pH 7.45, which is the physiological pH. So, it's the same principle for bases as with acids. So basically, a base is a molecule which can accept a proton from solution. Okay, so we start off with lysine, which is our base. Then once it has actually accepted a proton, it's no longer a base because it can't accept another proton. So in fact, it's then called the conjugate acid because in fact it's an acid because it can donate that proton which it's received, this proton here, or wet back into solution basically. Okay, right. So the important thing to understand is that if you have a whole bunch of basic residues in this apolipoprotein B100, at any one time a good fraction of those will be protonated and therefore will be positively charged. So basically the message is that the type A repeat domain of the LDL receptor will be negatively charged and the apolipoprotein B100 will be positively charged, and that's how these two interact. Okay, and that will be important later when we come to discuss how the two cleave apart within the late endosome. Okay, right. So, then what will happen is the binding of the LDL to the LDL receptor will trigger a conformational change in the LDL receptor, which will lead to the exposure of an endocytic motif on the cytoplasmic side. Now, the endocytic motif is basically a sequence of 4 amino acids which tells cytoplasmic proteins to begin the endocytosis process, basically. It marks this protein out as something that should be endocytosed, basically. So what is the endocytic motif that the LDL receptor has? Well, basically, there are many examples in biology of proteins which need to be endocytosed, and they all have different endocytic motifs. The one that the LDL receptor has is something known as the NPXY motif. Okay, and to understand what this means, you need to be able to read single letter amino acid code. Okay, so basically, we're using single letter amino acid code here. N, the first amino acid within this endocytic motif, stands for asparagine, okay, which is the primary amide of aspartic acid. P, the second amino acid in this endocytic motif, stands for proline, okay. Little x does not stand for any amino acid, it just means you can have any old amino acid in that third position, so we're not bothered about what's there. And then in the fourth position, Y is the single letter amino acid code for tyrosine. Okay, so this is the sequence of amino acids that you need to have present somewhere within your cytoplasmic portion of your protein that needs to be exposed so that other proteins can come and bind to this endocytic motif and trigger the endocytosis of this LDL receptor, which has got the LDL cargo bound to it. Okay, so I think it would be quite nice to actually draw out this endocytic motif. Okay, so let's start with asparagine. We don't really have space here, so I'll move down to here. So, asparagine then. Here is the amino terminus of this asparagine amino acid. Here is the alpha carbon with a hydrogen coming off it, and here is the carboxylic acid group. Okay, the R group then of an asparagine amino acid, which is this first amino acid in this endocytic motif, has a methylene group and then a carboxylic acid group, which is now involved in a primary amide link, and therefore instead of having the alcohol group coming off here, it instead has an amino group coming off there. Okay, so this is asparagine, or N. Okay. Then the second amino acid within this endocytic motif is going to be proline, and again we'll draw the structure of this out. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group, so that's the core structure. Now, in the case of proline, your R group is actually going to involve the amino group, so what I'm going to have to do is I'm going to have to remove this hydrogen here. Okay, so cross that out. That's not going to be there in the case of proline. Instead, what you're going to have is an R group that is a 5-membered ring which involves this nitrogen atom here. Okay, so I'll draw 3 methylene groups here. So these CH2s, those represent carbons with 2 hydrogens coming off. Okay, and then down here you have your next methylene group. So we've got a carbon, carbon, carbon forming this 5-membered ring between the alpha carbon and the nitrogen of the amino group, and then off each of these carbons you've got 2 hydrogens basically. Okay, and this is the structure of proline, or P. Then the next amino acid, we're not fussy about what this is. This can be any old amino acid. So here's its amino terminus, here's its alpha carbon, and here's its carboxylic acid group, and we'll just put an R group there to say that you can put whatever you want basically there. Okay, so this is a little X, and then finally we've got tyrosine in this final position here. So here's its amino group, here's its alpha carbon with a hydrogen coming off it, here's its carboxylic acid group, and then the R group of tyrosine consists of a methylene group, like so, with a benzene ring coming off the methylene group. Okay, and then this benzene ring has, on the opposite side, an alcohol group coming off it like so. So this is the amino acid tyrosine. Okay, right, so that's what is meant by the MPXY motif. You have this at some portion of your cytoplasmic portion of your protein, and this will become exposed when the LDL binds to the LDL receptor. Now what's going to happen is a special adapter protein is going to come and bind to this endocytic motif, and that's going to trigger the entire endocytosis of the LDL loaded LDL receptor. Okay, so let's show this now. So let's draw out our picture of the LDL receptor again. Okay, so here is the LDL receptor then. Here is the type A repeat domain extracellularly, and we'll now show this important motif that is now exposed here, which is this NPXY motif. Okay, so previously it wasn't exposed, and that's why the LDL receptor wasn't being endocytosed before it bound the LDL. Okay, so now here is our LDL protein, or our LDL particle rather. Okay, this is the phospholipid monolayer, and here we've got our apolipoprotein B100. So this is apo B100, and I'll colour in apolipoprotein B100 in orange. Okay, and it's bound to the type A repeat domain of the LDL receptor. Okay, right. Now what's going to happen is once the endocytic motif is exposed, another protein is going to come and bind to that endocytic motif. Okay, and this is called the LDL receptor adapter protein 1, or the LDL R for receptor, and then AP for adapter protein, and then 1, like so. So the LDL rap 1. Okay, so this stands in full for the LDL receptor adapter protein 1, and this is aptly named because basically this is going to bind to the LDL receptor once it's got its cargo bound, okay, and it's also going to bind to the Clathrin triskelions, which are the main endocytic machinery that's going to allow us to endocytose the LDL receptor with its cargo bound. Okay, so really it is an adapter protein because it binds to the two things and it adapts the signal, if you like. Okay, right, so we'll colour in the LDL receptor adapter protein 1 in blue here. Now, I should also mention that the LDL receptor adapter protein 1 has another name. It's also called the Autosomal Recessive Hypercholesterolemia protein. And this is because of its involvement within autosomal recessive hypercholesterolemia. In autosomal recessive hypercholesterolemia, basically you have no functional LDL receptor adapter protein 1. So in your two genes for the LDL receptor adapter protein 1, which I'll draw here, okay, so here are your two genes for the LDL receptor adapter protein 1, let's say. If you have loss of function mutations in just one of them, and then the other one's fine, then you do not get any disease symptoms. However, if you lose both of them, then you cannot endocytose LDL receptors, basically, when they bind that LDL, which means that your cells can't remove LDL from the blood, which means LDL just goes up and up and up and up in the blood. And of course, the LDL particles are laden with cholesterol. So this means that the amount of cholesterol you have within your blood has gone up, basically. And this is why it results in hypercholesterolemia. And it's not a nice disease to have. It's usually quite fatal early on in life. Okay, so they usually die very prematurely. Now, this protein was therefore found to be involved in this disease called autosomal recessive hypercholesterolemia, and was therefore named the autosomal recessive hypercholesterolemia protein, or for short it's often abbreviated to ARH. So you might see people calling this LDL receptor adapter protein 1 instead the ARH. Okay, right, so the LDL receptor adapter protein 1, or the ARH, binds to the MPXY endocytic motif on our LDL receptor. Okay, I'll just label up our LDL receptor here. Okay, and now what's going to happen is the LDL receptor adapter protein 1 is going to bind also to Clathrin triskelions. Okay, so let me discuss with you the structure of a Clathrin triskelion. Okay, so I'll start with the structure of Clathrin then. Okay, so basically Clathrin is made up of a Clathrin heavy chain and a Clathrin light chain. So let's start with the structure of the Clathrin heavy chain. So basically the Clathrin heavy chain has a structure that looks like so. This portion right over here, okay, and I'll highlight this in. Okay, so this portion that I'm now highlighting in blue is called the N-terminal domain. Okay, so this is called the N-terminal domain. This portion here, which I'll highlight in green, is called the distal domain. Okay, so we've got the N-terminal domain right at the end here, and then we've got the distal domain following that. Okay, and then we've got this larger domain in the middle, and we're going to split this into two portions. Okay, so the first portion, which I'm highlighting now in pink, is going to be called the proximal domain. Okay, so the portion in pink is the proximal domain, and then after that, the portion that's going to be right in the center of the Clathrin triskelion, when we see the Clathrin triskelion, this portion, which I'll highlight in red here, which wasn't a great decision because it probably won't show up that different to the pink. This portion is called the trimerization domain. Okay, and it's going to be involved in trimerizing 3 Clathrin molecules together to make a Clathrin triskelion. Okay, so this is the trimerization domain. So this is the Clathrin heavy chain so far. We have not seen the Clathrin light chain. This is just a half of a Clathrin molecule, the Clathrin heavy chain. Then attached to the Clathrin heavy chain, specifically the proximal domain of the Clathrin heavy chain, you then have a smaller protein, which is now here in turquoise, which is called the Clathrin light chain. And when you have this Clathrin heavy chain associated with a Clathrin light chain, like so, the entire molecule is then called a Clathrin molecule. Okay, so this is the structure of Clathrin. Now, in the cell, Clathrin doesn't usually exist like this. Instead, what happens is these Clathrin molecules are going to trimerize to make Clathrin triskelions. Okay, so let me now show you the structure of a Clathrin triskelion. So basically, if we redraw out our Clathrin molecule, here's our Clathrin heavy chain associated with a Clathrin light chain. What we're going to do is take 3 of these and bind them together. Okay, so here comes another one. So this is another Clathrin heavy chain coming off here. Okay, and they're trimerized together, they're joined together at the trimerization domains here. So I think I'll just colour in the trimerization domains. So those are the 2 trimerization domains of these 2 Clathrin heavy chains. And then this Clathrin heavy chain will have a Clathrin light chain associated with it as well. But it's a trimer, so we've only joined 2 of them together now, so we have another one coming off here. Okay, and I admit that this does look rather like that horrible symbol from history. Okay, but this is what the Clathrin proteins do look like when you look at them in the cell. Okay, so this is a Clathrin triskelion. Okay, right, and they're all joined together by the trimerization domains of the Clathrin heavy chains, which are at the centre. Oh, and I've missed one little bit off, which is that light chain that will be associated with that final heavy chain. Okay, so this is how you find Clathrin present within the cell. Now, what will happen is once the LDL receptor adapter protein 1 binds to the endocytic motif of the LDL receptor, which has LDL bound to it, what will now happen is a Clathrin triskelion will come and bind to this. Now, I'm not going to attempt to draw the Clathrin actually as a triskelion as I've shown here, but instead I'll just show it like so. So this is a Clathrin triskelion here. Okay, coming and binding, and then what can happen is other Clathrin molecules can bind to this first Clathrin molecule, and they can start to polymerize into a buckyball. Okay, so let me discuss the structure of buckyball with you and how Clathrin triskelions can assemble into a buckyball-like structure. Okay, so basically a buckyball is a 60 well, it's a shape that has 60 corners, basically, and it's a 3-dimensional shape made out of tessellating hexagons that are regular hexagons and regular pentagons as well. Okay, so if you are familiar with the structure of a football, that's the structure basically of a buckyball, or a soccer ball if you're American. Okay, so that tessellation of regular hexagons and regular pentagons that makes up the soccer ball, that is what these Clathrin triskelions are actually going to associate into. So I'll attempt to draw a little picture of one of these. It's difficult because obviously they are 3D and I've only got 2D to draw in. So imagine here, this is going to be my first pentagon of the buckyball, okay? And imagine that that's in the plane of the piece of paper. Then what you can imagine is, we'll say this is the top of the buckyball, so I think it usually works better if I try and draw it as though we're drawing the top and it's going into the page rather than trying to draw it coming out of the page. Okay, then what I'm going to do is attach regular hexagons onto the sides of these pentagons, basically. And these regular hexagons are not going to be sitting in the same plane as this as this pentagon. They're going to be folded in and going into the page, basically. Okay, so let me show this like so. So here is my regular hexagon, and it's meant to be going into the page, so don't think of it as being in the plane of the paper. Imagine it's bent inwards, basically. And then we're going to have another one like so, here. And those two sides, the sides of this hexagon and the side of this hexagon, those will be joined together, and that's why they have to be bent inwards, because if they were in the plane, you'd have a gap between them, a little wedge between them. Okay, like so. So imagine we're starting to form the top of a dome that's going into the page here. Okay, here's another hexagon here, and then I've got another hexagon here, and the final one here. So you've got those 5 hexagons, and they're all bending back into the page. Okay, and then in these gaps here, these are going to be the tops of another pentagon. So what we're going to do is put in another pentagon here, and these pentagons will almost now be vertical. They'll almost be perpendicular to the plane of the piece of paper. Okay, here is another pentagon here, another pentagon here, another pentagon here, and then another pentagon here. And then what you'll do is around each of these pentagons you'll put 5 hexagons. You've already got 2 in each case, but you'll then put 5 hexagons, and the whole thing starts to fold round, and it works absolutely perfectly. So if you're really enthusiastic, what you can do is cut out regular hexagons and regular pentagons that all have the same side length, and you can try and build one of these things if you want. Mathematicians generally do it because they need it for group theory, because the symmetry groups of these things are very interesting. But if you don't understand the structure of a buckyball, I would recommend, to get some intuition, actually making one. Okay, so this is a buckyball basically, or just look at a football. Okay, now it's the structure then of Buckminster fullerene, which is a carbon structure where you have carbon atoms in all of these positions. Okay, so what we want to see is how do Clathrin triskelions assemble into one of these things? Well, the answer is not perfectly. Okay, it would be absolutely beautiful if these things could just somehow assemble into one of these things with absolutely no overlap, but there is going to be overlap. Okay, so you're not going to have no overlap between Clathrin triskelions. So let me show what I mean by this. So let's say we're going to put in a Clathrin triskelion here, basically. Okay, so this is going to be the center, where I've highlighted in blue, that's the center of the Clathrin triskelion. Okay, so here is one Clathrin heavy chain, here is the second Clathrin heavy chain, and here is the third Clathrin heavy chain. And of course all of those will have light chains associated with them, so let's put those in vivid purple here. So there's one Clathrin triskelion. Okay, now maybe we'll center another Clathrin triskelion here, and then it will overlap basically. So if we center another Clathrin triskelion here, I'll colour this one in green then this time, then we'll have one of these Clathrin heavy chains going off like that, and then this second one will come off and overlap completely there, and then this one will go off like so. So basically the Clathrin doesn't not overlap. It does have overlaps when it's building one of these buckyballs, but hopefully you can appreciate how you could build the skeleton of one of these things out of joining together loads of Clathrin molecules, and this is what starts to happen. So you bind the first initial Clathrin molecule onto the LDL receptor adapter protein 1, and then it starts binding with loads of other Clathrin proteins, basically, loads of Clathrin triskelions, and it starts assembling one of these buckyballs at this point where the LDL receptor is. So let me explain the significance of this. Basically what's going to happen is you're going to start forming an endocytic vesicle which has this LDL receptor in, like so. Okay, and let me explain why. So we have our LDL receptor here. Okay, so here's the type A repeat domain of our LDL receptor. We then know that we have this important domain that's on the extracellular side, which is just 4 amino acids, but because it's so important, I'll draw it enormous, like so. Okay, which has bound to this LDL receptor adapter protein, and because I haven't got much space, I'll just call it the ARH, the Autosomal Recessive Hypercholesterolemia. And now what's happened is that a Clathrin protein has bound to that, and then the Clathrin protein is starting to form one of these buckyballs. Okay, so it's starting to polymerize with many other Clathrin proteins, and it's forming a buckyball. Now I'm not going to attempt to draw the buckyball here, okay, but what I will draw is this structure like so. So these are the polymerizing Clathrin molecules that are forming this sphere shape, basically, which is now sort of pulling the membrane downwards into this ball shape, basically. Okay, so this is multiple Clathrin proteins all bound together here. Okay, so I'll separate them all off. So let's colour in the Clathrin proteins in a certain colour, and I think I'll go for blue as the colour to show them. Okay, so all of these proteins that I'm now colouring in blue, these are Clathrin proteins. Okay, and then they are pinching off this vesicle, basically, which is going to contain the LDL receptor here, and I haven't actually drawn the LDL bound there, but the LDL will still be bound there, basically. Okay, so here is our LDL particle, which has the apolipoprotein B100 in there, bound to the type A repeat domain. Okay, so let's colour everything in. So we'll have the apolipoprotein B100 in orange here. Okay, we'll have our LDL receptor in green. Okay, like so. We'll have the autosomal recessive hypercholesterolemia protein, or the LDL receptor adapter protein, in vivid purple here, and then it's bound to the Clathrin, which is now polymerizing to form a buckyball, basically, like so. And then what will happen is it will end up completely pinching the vesicle off. So what you'll end up with then is an endocytic vesicle, okay, here, which is a little membrane-bound structure with a phospholipid bilayer. That's important, that this membrane that we've got here is a phospholipid bilayer in itself. Okay, and this endocytic vesicle contains the LDL receptor here, okay, with the LDL bound to it, like so. So here's the phospholipid monolayer of the LDL particle with the apolipoprotein B100. Okay, so let's continue our story. And here is the apolipoprotein B100 in orange, and the LDL receptor again I'll draw in green, so that we have some consistency here. And then we've got, remember, our MPXY motif on the intracellular aspect here. Okay, and then we have the autosomal recessive hypercholesterolemia protein here, or the LDL receptor adapter protein 1, and then this is associated with Clathrin, and the Clathrin has now completely formed a buckyball around this. So let me try and show this. Okay, so here is our complete buckyball around the entire structure. So at the moment this will be called a Clathrin-coated endocytic vesicle. Now, before the endocytic vesicle can do anything, it needs to shed this Clathrin coat, because whilst it has this Clathrin coating it like this, it can't actually interact with anything. It can't fuse with another membrane-bound organelle within the cell, and it needs to, basically. It's going to go to an intracellular organelle known as the early endosome. Okay, and to fuse with the early endosome, it needs to shed this Clathrin coat. Okay, right, so let me colour in this Clathrin coat in blue here. So this is a Clathrin-coated endocytic vesicle, and again, although I have drawn this Clathrin coat very simply here, like so, in reality it would be a buckyball of these Clathrin proteins, like I've previously discussed. Okay, so here is the Clathrin coat shown in blue. I'll separate all the different Clathrin proteins. So many different Clathrin triskelions have joined together to make this Clathrin buckyball, which is surrounding the endocytic vesicle. Okay, right, so this is now in the cytoplasm of our hepatocyte, and what has to follow next is the uncoating. So what's going to happen is the Clathrin particles are going to be split off from this endocytic vesicle, okay, and I think I'll get another piece of paper for this. What's going to happen is also the autosomal recessive hypercholesterolemia protein will part its ways with the LDL receptor as well. So what you'll end up with then is a vesicle like so in the cytoplasm of the cell, okay, which has the LDL receptor bound to it, and I won't bother showing the endocytic motif anymore because it's done its part now, okay, and also an LDL particle bound to it like so. So here's the LDL particle with this large apolipoprotein B100 bound to the type A repeat domain still of the LDL receptor. Okay, that's where we've got to at the moment then. So this is known as an endocytic vesicle. Now I want to stress something very important. This is not an endosome. Okay, there is a difference between an endocytic vesicle and an endosome, and they are not the same thing. An endosome is a massive, great intracellular organelle. An endocytic vesicle is one of these tiny little spheres of membrane that we've pinched off from the cell membrane. Okay, so to put this in perspective, if we've got our entire cell here, an endosome is a large intracellular organelle like so, whereas an endocytic vesicle is a tiny little blip, basically like this. And what's going to happen is this endocytic vesicle, which has come from the plasma membrane and which has this LDL particle within it, is going to now go to an endosome. Okay, and the first endosome it's going to go to is something called the early endosome. Okay, so this is the first endosome that it's going to go to, and an endosome is a large intracellular organelle. Okay, so what's going to happen then is if we draw this here, here is the early endosome, and what's going to happen is the endocytic vesicle is going to come and fuse with the early endosome, and then what will end up happening is that the LDL receptor will then end up in the membrane of the endosome. Specifically, this is the early endosome at the moment. Okay, and it will also have this LDL particle bound to it. So let's show this here. Okay, so here's the LDL particle. Here is the apolipoprotein B100, which we'll have in orange. Now, very little is going to happen in the early endosome. And what's going to happen is it's going to get shipped off very quickly to the late endosome. Okay, but I think we'll discuss that in the next video.